thank very much the organizer for inviting me to present some results that my group is been getting in the last 5 years when I based my research group at the Biotechnological Institute at INTA. As the title of my talk is tackling the metabolism of important nutritional component in crop species, I would like to introduce you to the model that we are using in my group for boarding the metabolism, the plant metabolism. Just a couple of years ago, when I was doing my postdoc at the Max Planck Institute of Plant Molecular Physiology, I became interested in the plant metabolism and particularly in the tomato fruit. This is important because one of the feature of this system is that you have to deal very early in the development of this organ with an autotrophic metabolism, even when this is still a matter of debate. And immediately after, in a very short period of time, you get into an heterotrophic metabolism. This makes the tomato fruit very interesting for a model to board the metabolism. And also, the other interest is that the tomato fruit is a very important source of nutritionally important compounds that the human diet is composed by. And the first change that you observe in the metabolism of tomato fruit is the obvious changes in the color that are made by the differences in the pigment and how the pigments are changed from the very beginning development until the fruit is mature. And in 2003, 2005, at the Alistair Fernie laboratory, we set a platform based on GCMS to rapidly detect and quantify about 70 polar metabolites in the tomato fruit in different stages of development. And we observed many, many differences in the content of the different metabolism of the carbon and primary metabolism. And we reasoned that if these differences are becoming so rapidly, we will be able to somehow engineering the tomato fruit to produce, to enhance for nutritional compounds in this fruit. The other things that we thought is that the metabolic variations in the tomato fruit can be better exposed when we look at the native related wild tomato species. And this is important, especially at the regional level, because the different species, which are enormously diverse, are all of them, or almost all of them, native from the South American region. Actually, when Alistair Fernie did the experiment trying to search for differences in the metabolite contents in the different fruit of these species, they found a tremendous variation in the metabolite contents related to amino acid, organic acid, sugar, some phosphate intermediate, and some secondary metabolites. This is why we board a very large project trying to expose the genetic determinants of this metabolic variation in tomato. And for this, what they have used were an introgression line population composed by Solanum pennelli and Solanum lycopersicum. The Solanum pennelli is the very close related wild species, and Solanum lycopersicum is the domesticated variety. And even when they are highly divergent, these species can be crossed and they produce viable progeny. The systems that they used to expose this variation and also to try to expose the genetic determinant were 76 different introgression lines derived from this cross. And actually what they found was many, many different loci associated to quantitative variation in the metabolite content along all the introgression lines and in about these 70 metabolites that I mentioned before. Just to explore the genetic determinant of this variation, the first approach that we took was to explore the Solanum pennelli genome And when we started with this project, there were almost nothing related to wild species, related to the sequence of these wild species. What we did first was just to try two different approaches, one related to candidate genes, where we look at the different genomic region for already described genes and mapped on this region. And the second approach was to construct physical mapping of this interesting region that we detected before. Just briefly, what I wanted to comment on this result was that exploring the Solanum pennae genome allowed to find that although by compression of the domesticated species, the Solanum lycopersicum species, the compression of the genome of this species, both genomes in the region that we have analyzed are clearly linear, and the synteny is maintained in different regions that we analyzed. This happened both at the microsynteny and also at the microsynteny level, where the genes at the intergenic region are interrupted by the insertions or deletions of different transporter or transporter relic that are still there. This is the reason because we thought that boarding the sequencing of the entire Solanum pennae genome will be a great resource for exposing the genetic determinant that we are looking for. In this way, in a small consortium of 5 labs from different countries, from the States and also from Europe, we boarded the Solanum pennae genome, and at the moment, this project is almost complete, where we got more than 80% of the sequence of this wild species, and the N15 scaffold length is a very good number for our purpose, and also the sequence that we got are at the moment 40% anchored to the different chromosomes of the tomato. With this research in hands, in the next few minutes, I will show you some results that are coming from the last 2 or 3 years of work in my group, where we zoom into QTL for nutritionally important compounds. In this case, we zoom into QTLs that were detected before for the content of vitamin E in fruit. The vitamin E, I have to introduce to you, the vitamin E compounds are composed by 2 kinds of compounds, the tocopherols and tocotrienols. They differ in the methyl group positions of the chromanol head, and they occur in the nature in 4 forms: alpha, beta, gamma, and delta. They are liposoluble antioxidants and are essential for human diets. They are synthesized at the inner chloroplast envelope and also in a very ultrastructure of the chloroplast, the plastoglobule, and they can be easily detected and quantified by liquid chromatography. The questions that we asked when we board this project were how vitamin E synthesis is regulated in tomato, how it moves intracellularly, if it does, and which is the role of the tocopherol in green organisms, and how this pathway evolved in higher plants. Thinking in this question, the first experiment that we did were just to measure the different form of tocopherols in different introgression lines that I mentioned before, and compare with the Solanum lycopersicum content in the mature fruit. As you can see here, we detect several QTL at chromosome 6, 7, 8, and 9. These QTL are related to are QTL for the different form of tocopherol that we could detect at the tomato fruit when they mature. What we did next was to try to reconstruct the pathway that lead to the vitamin E biosynthesis in tomato. What we did was just localize and clone all the genes for all the enzymes that are working in the tocopherol biosynthetic pathway, this blue box marked here, and also the 2 divergent pathways, the methyl erythritol pathway and the shikimate pathway, which provide the intermediate for the final product tocopherol. Next, what we did was just to map all these genes in the in tomato, and you can see here that except in chromosome 12, of all these genes marked in green, those derived from the related to the shikimate pathway, in red the MEP1, and in blue the tocopherol core pathway. They are distributed all along the genome and somehow randomly. If we come back to the QTLs that we have detected in these 4 chromosomes, we can localize or co-localize the different genes that we have mapped together with the QTLs related to the vitamin E content. What we did next was to set a quantitative PCR array for study the transcription or the mRNA accumulation of all of the genes related to these 3 pathways. In this picture, what we can observe is the variation in the transcript level for the different enzyme encoding genes related to the methyl-erythritol pathway, the shikimate1, and the tocopherol core pathway. What we can see here is the variation in the transcript level suggests that, at least in part, this variation could explain the variation in the tocopherol content, at least when we measure the transcript along the fruit development and also in photosynthetic tissue as in SIRS and zinc leaves. If we take all this data and try to see how this data explain the different developmental stage of the tissue that we have analyzed, this picture emerges. And also, if we take all the eigenvector of this analysis and reconstruct, or even better, or better to say, construct a network analysis of this eigenvector, we can see 2 clearly differentiated networks. One, which is comprised of those eigenvectors that can separate fruit from leaves here, and the second one, which can separate fruit and leaf from immature fruit here, which is this small network. If we concentrate our attention in this network, we see that the most of the, or even better, the most connected genes in this network are those related to the core pathway. If we took another approach as a proof of concept of this previous analysis, what we did was just to collect different tomatoes varieties from different places in South America. We measured the tocopherol content, the vitamin E content of these varieties, and also the pigment content, and also we measured the expression of the genes related to the tocopherol biosynthesis. What we have identified were a couple of genes, or few genes, which can explain, or somehow explain, or correlate well, better to say, the differences in the total tocopherol contents. This result let's see. Oh, here we are. Moreover, what we did next was to try to understand this variation in the transcript level of the genes by searching for cis-regulatory motif in the tocopherol genes in tomato. We apply a novel method to detect the novel cis-regulatory motif in the promoter region of the genes, and also we apply the same method for searching this motif in 3 different Solanus species. And finally, what we did was just to study the co-occurrence of this de novo identified cis-regulatory motif with the transcript profile. And what we found was that the motif are differential distributed among the toco gene. Also, we found that the motif in toco genes are differentially distributed among the species. And finally, we found that the co-occurrence of this motif correlate well in some instance with the expression pattern of the genes. This result, and the previously mentioned in the previous slide, slide lead us to propose regulatory points along these pathways related to the entrance to the main intermediate for the MEP pathways and the entrance to the intermediate or the substrate for the chocophyll biosynthesis, which are mainly the fetal availability, the fetal 2-phosphate mediated by BT5, and this step mediated by the DXS enzyme. Also, these three genes related to the core pathway seems to be central in the regulation of the chocophyll content in tomato fruit. If we come back to the previously mentioned QTL, let's call this QTL BT9QML, this QTL colocalized with these genes in the central chocophyll pathway of chocophyll biosynthesis. What we did was just to cross this introgression line, which contains the introgression, a big introgression containing the Solanum pennellii allele, again back with the M82 variety, the Solanum lycopersicum variety. We produce an F2 population, and when we measure the chocophyll constant in this F2 population, we detected that the Solanum pennellii, the lines carrying the Solanum pennellii alleles and also the lines in heterozygous status, they have an increase in the relative contents of the different form of chocophyll. This happened as well with the content of another antioxidant in the plant. When we explore the difference in the alleles, in the BT3 alleles, we found not so many differences at the sequence level and neither to the protein level. We proved that this protein is targeted to the chloroplast here in this with this GFP fusion. We also explore and we also confirm that the BT3 is regulated along fruit development, and more importantly, we found that this QTL is an expression QTL for these genes. Actually, those introgression lines that are carrying the pennellii allele show between 2 and 3 higher level of transcription for these genes, both in leaves and in different fruits. When we silence these genes, specifically in tomato fruit, what we observed was a clear reduction, clear differences in the chocophyll content of the silenced fruit. And also, when we also found antioxidant capacity altered in this silenced fruit, with a mild effect in central carbon metabolism. The next question was whether the mechanism of this QTL, whether this is transcriptionally or post-transcriptional regulation in this gene. As mentioned before, what we looked were for different motifs in the promoter, but these results in the differences in the motif in the promoter could not explain the difference that the expression that we observed. Actually, the